In this video, I'll be discussing human blood groups, how you can identify them, and then how you can actually explain what kinds of blood types can be donated between individuals. To begin with, we're going to look at what are known as antigens of the human blood groups. There's more than one type of blood group. We're going to focus here first on what's known as the ABO blood groups. And as noted here, these are determined by the hereditary or genetically induced presence or absence of antigens A and B on an erythrocyte surface. So an antigen to begin with is a glycoprotein usually that comes right off of the surface of the plasma membrane. And it is an identifying marker of some kind, and each cell has many types of antigens, identifying everything from where it should exist to what it can interact to what can bind to it. And so these antigens we're referring to are just a couple of many antigens that you would see on any particular cell, and specifically here, erythrocytes. And so to begin with, these antigens themselves are only labeled as antigen A or antigen B. Now, since each individual genetically has copies of genetic information from mom and dad, what that means is they would have two alleles or two tiny genetic components that code for what their blood type is. And so here in this short table, you can see here that if an individual has no antigens on their surface, then that's because the two genotypes they receive are not designated as A or B. And so this is what's characteristic of type O blood. So then, of course, if you come over here and you see somebody with the antigen A, just one antigen A, that could be because both alleles that this person receives are demarcated as A alleles, or they could have just one A allele and one allele that codes for no antigen. Likewise, same thing can happen with somebody with a B antigen. It's because either both alleles they receive could have B antigen genotypes, or they could be a combination of the B antigen and then, of course, the genotype or allele that designates for nothing. Now, finally, you can have a combination of the two. So an individual that receives an allele for A antigen and an allele for B antigen ends up with both antigens present on the surface of their cell. And so down here, we're going to begin to look at what these erythrocytes would be classified as based on the antigens that are present. And on each one of these cells, right, so this would be one erythrocyte, and off of that I've drawn what would be one antigen. So this is, of course, showing you an A antigen on the surface of this cell. I recommend that anytime you begin to discuss a blood type, that you sit down and first draw a round erythrocyte, and off of it, draw each type of antigen that you would expect to see. This is going to be really helpful for learning to classify visually what blood types you're working with. And we're going to go through and finish classifying each of the visual examples at the bottom, but before we do so, I want to talk about the second blood group we used. And that's going to be the Rh blood group. And so this is going to include a number of antigens, C, D, and E, but we specifically look at D since it's the most important in terms of transfusions. And so what we see here is the presence of a D antigen on the surface of an erythrocyte can designate this ABO blood group as a positive. So you may have heard of a blood being a positive or a negative blood type, and positive indicates that D antigen is present. An absence of it indicates that blood group is going to be negative. And so what you want to do here is be able to look at each of the examples I've given you here and diagnose, essentially, what that blood type would be. So let's walk through them all. So a person here with only an A antigen, right, but no D antigen would be known as A negative blood type. Now, if you take that same individual right here, or a similar erythrocyte, and you give them an A antigen, and then you also add a D antigen on that cell, that's an A positive blood type. So again, that Rh factor, right, also known as the D antigen that we're looking at there, is either A negative or A positive depending on its absence or presence. Of course, then we slide over here, this third example, it's showing you a B antigen and an A antigen, but no D antigen. So you could ask yourself what kind of blood type that would be. And that is AB negative. Again, A and B because both antigens are present, but negative because there's no D antigen. Come over here and you see a B antigen and a D antigen. That's going to code for B positive blood. Now, like we said earlier, it is possible for an erythrocyte to not have an A or a B. And based on what you saw in this table, having neither of those antigens dictates you as type O blood. Now, you can still be type O blood even if you have a D antigen on the surface, and that would be O positive blood. And so O negative blood, your naked cells essentially, are the ones that don't have any of the A or B antigens, and they also don't have the D antigen. So that's the basics there. Again, the first thing you should do with any blood type is be able to draw the antigens on the surface of that erythrocyte. Now we take it one step further and we look at the type of antibody that an individual would be producing based on what kind of blood group they make. So to define this, an antibody is a gamma globulin, and that's going to be secreted by immune cells. The goal of an antibody, as it says here, is to bind to foreign antigens. This is any foreign antigen on any cell, and mark them for destruction. This is not a good thing. Anytime an antibody binds to something, as it says here, it can cause clumping, or what we know of as agglutination of these foreign cells. Now, this is what we would refer to as a transfusion reaction if somebody receives a blood type that their antibodies recognize. So if your antibodies attack a blood type that you've been donated with, right, or transfused with, that causes those cells to clump, and that slows blood flow, and that can be lethal. So as we look specifically at the antibodies associated with blood type, it's important to note here that antibodies of the ABO blood group react to any A or B antigen except their own. Now this is key. You do not want antibodies that attack your own cells. That does happen in some cases, and that's what we know of as an autoimmune disease or an autoimmune reaction. That is not the norm, and so you don't want to be assuming that a person's ABO antigens, again on the surface of their cells like we saw down here, those are not the same as the antibodies that we see here. Now, by name, we see the first antibody is known as alpha agglutinin. This is also called just anti-A antibody. This is named because it will react to antigen A. So to put it simply, anti-A antibodies bind to antigen A on the surface of erythrocytes. Likewise here with beta-agglutinin, this is your anti-B antibody, and this was going to react with antigen B to induce agglutination. It literally hunts down antigen B on the surface of cells and binds to it and eliminates those cells. The last thing you see here is anti-D antibodies. So remember, the D antigen right here is what was going to dictate positive or negative blood types. And so you do have antibodies for them, but these are not normally present in the blood. They only form when an individual who is Rh negative, such as this O negative, or this AB negative, or this A negative person, right? So they only form if one of these Rh negative individuals are exposed to a D antigen. For example, maybe they receive a blood transfusion with positive blood type. At that point in time, those individuals will start to make these anti-D antibodies. Otherwise, the alpha agglutinin and the beta agglutinin are the only antibodies you would see prior to a reaction. And so we continue to look at the same table we started earlier. For type O blood, we find that these individuals do innately carry anti-A antibodies and anti-B antibodies. If you're a type A individual, you are going to carry anti-B antibodies in your serum. Type B erythrocytes will be coupled with anti-A antibodies. And of course, if you have type AB erythrocytes, right, again, one of these right here, where they have A and B antigens, they won't have either of those antibodies. And so what's really difficult here is starting to understand visually the relationship between the two. So I've taken those same blood types that we studied a few seconds ago, leaving their names here below, and your goal is to begin to write in the kind of antibody that would be present in their serum. Okay, and so each box represents one person. And each of the individuals here, all 6 individuals, have different blood types. So if we start here with this individual who is A negative, you ask yourself, would they be making A antibody? Well, if they do, the A antibody is going to bind to their A antigens, causing agglutination of their own cells. So that's a no. The only kind of antibody this person makes is an anti-B agglutinin. Now, what that means is twofold. First off, if this individual receives erythrocytes from somebody else that happen to have B antigens on them, those B antibodies will bind to those cells and clump them together. But as long as that doesn't happen, then this B antibody will never attack the A antigens on the surface of this person's cells. And so they're safe. Now, likewise, this individual is Rh negative, but as we said a second ago, anti-D, right? Antibodies are not normally present in the blood. This person would only start to have these if they were exposed to D antigens for some reason or another. So let's keep going with this. So an individual here that has A positive blood, once again, ask yourself, would they have A antigen antibodies, B antibodies, or neither? Right, and the answer is they are also going to have anti-B antibodies. They don't want anti-A because it would attack their own cells. Now, this person having positive blood will most definitely not have anti-D antibodies because in that situation, that anti-D would also attack this individual's cells. So anti-B antibodies are the only thing this person is going to make. We move on then. Now, this individual has both B and A antigens on the surface of their cells. And so you won't find that they have either of those antibodies. If they had anti-B antibodies, it would attack their B antigens. And if they have anti-A antibodies, it would attack these A antigens here. This individual happens to be known as a pretty universally accepting recipient. It's not completely. In fact, only AB positive is the true universal recipient. But this person here can accept a lot of different blood types just because they're not making any antibodies innately. Now again, because they have a negative blood type, if they were to receive cells that have D antigens on their surface, then they would start to make anti-D antibodies. And so let's move down this way. This individual has B positive blood, so the only antibodies they will make will be your anti-A antibodies. This individual has O positive blood, so you'll notice there's no A antigen and no B antigen. Since neither of those are present, this individual will carry anti-A antibodies and anti-B antibodies. If you think about it, this individual really can't receive a wide variety of blood types, right? And that's because he can't receive anything that has A antigens on the surface, like this, or this, or this, because those A antibodies will immediately attack him. Likewise, because they make B antibodies, they can't receive any blood types that have B antigens on the surface. Otherwise, they would attack that. Again, this is a positive blood type. It does have D antigens on the surface, and so this individual is not going to be making anti-D antibodies. However, once you make it over here to this O negative blood type, you see that they start off by making anti-A antibodies and anti-B antibodies. If they were to come in contact with D antigen, for example, if they were to receive an A positive blood type with its D antigens, or if they received O positive blood type with its D antigens, then they're going to start to make that anti-D antibody. So that's the idea there. And so that's the second step in knowing how to work with human blood groups, is knowing how to classify which antibodies you would find in that person's serum. Now, blood transfusions themselves occur anytime blood is transferred from one person to another. A transfusion reaction will occur in certain cases to induce clumping and destruction of those transfused erythrocytes. And again, this occurs when an individual receives erythrocytes coated in antigens that can be attacked by their own antibodies. The downside here is that this widespread hemolysis and destruction of erythrocytes causes you to have reduced oxygen carrying capacity. And because of all of the clumping that's taking place, this slows down blood flow. Earlier in the notes, we talked about blood viscosity. This would increase blood viscosity and thickness. And so as we finish up looking at this table here, We've continued to talk about the genotype, the antigen, the antibody present, and now it's important to understand what kind of erythrocytes an individual can receive during a blood transfusion. So you can see that list here as you go through. And the downside to the list provided here in the textbook is it does not include positive and negative blood types or D antibodies, D antigens. And so this is just a jumping off point, but it's not a complete list. Now, ideally, you would want to have a blood type donated to you that is as identical as possible to your own. And in some cases, a patient can pre-donate their own blood, have it on reserve somewhere, so they can receive an autologous transfusion when in need. That way they don't have to worry about finding somebody with the same blood type. And so in this series of slides, we're going to go through and try to figure out whether or not a transfusion will work. On the right side of the screen, you'll see a recipient's blood type, and on the left side, you'll see a donor. All right, so I'll go through and I'll walk through it, and then I want you to pause the video and see if you can guess whether or not it will work. So to begin with here, the recipient has A positive blood. They have A antigens and D antigens on the presence of their cells, and their body innately makes B antibodies. Over here, you have an individual with A negative blood. They only have A antigens on their cells. The question is, if this donor transfers these erythrocytes to the recipient, will that be a successful transfusion? You can stop and ask yourself, if this erythrocyte is transferred to this recipient, will his antibodies attack? And the answer is that yes, this can be a successful transfusion. Because the donor cells only have A antigens on their surface, there's nothing for this B antibody to recognize and bind to. This is a safe transaction. We move to a second example here, where an individual with AB negative blood wants to donate to a recipient with A positive. This is the same recipient that we saw earlier, but now it is a different donor. And so the question is, will this donor's cells be recognized by the recipient's antibodies? So here what you would find is that yes, the B antibodies present here in the recipient will recognize the B antigens on the surface of the donor's cells. A transfusion reaction will occur, so no, this is not going to be considered a successful transfusion. Again, the issue here was not necessarily what antibodies were coming from the donor. Those tend to be so small in number, it doesn't matter. But what's important is whether or not the antibodies in the recipient will recognize and bind to the antigens from the donor. Here's another example for you. So the donor here wants to donate B positive blood, while the recipient wants to donate AB negative blood. Now, of course, you'll recall that AB negative individuals do not have any antibodies initially. And so because of that, this is a successful transfusion. This donor can safely donate B positive blood to that recipient because they lack antibodies. Now, I did include the donor's antibodies, anti-A, over here in their serum, but as you can see, not many of those transfer over. And so since it's not the antibodies that are transferring in large quantities, this can still be a successful transfusion. In another example here, you can see that this recipient, AB negative blood, is now trying to receive B positive blood. Right? And so we saw here that this individual could receive this the first time around. And this is because initially, this AB negative individual is not making anti-D antibodies. Okay, so let's assume that this particular transfusion works one time. Okay, that'll work one time. But let's assume that following that transfusion, you wanted to try it again. Now, in that scenario, now that this individual, this recipient, has encountered a positive blood type with D antigens on the surface, now this individual is going to start to make anti-D antibodies. And this is going to prevent future transfusions from actually functioning appropriately. Now that they are making anti-D antibodies, any erythrocytes that have the D antigen on the surface will immediately be recognized, bound to, and eliminated. And so while a one-time transfusion with positive blood to a negative individual can work, that recipient will start to make anti-D antibodies, preventing you from being able to donate positive blood types in the future. So no, the second time will not work. In order to be safe, it is just best not to transfer positive blood type donors to negative blood type recipients. So over here, we see one final example. We see instead of using O positive blood, now we're using O negative blood to transfer to this AB negative individual. And since they are making anti-D antibodies following that positive blood transfusion, that has nothing to recognize, so this would be a better choice. This is a more successful transfusion. So it's important to talk about what happens during pregnancy here. You do have scenarios where you can have negative blood type mothers, so Rh negative mothers, where they do become pregnant with Rh positive babies. This is because the placenta between babies and mothers does not allow whole blood to cross, but there are some antibodies that can cross and there are some proteins that can cross to cause reactions every once in a while. And so if an Rh negative mother is pregnant with an Rh positive baby, this tends to be just fine during that pregnancy. However, during delivery, there is sometimes tearing and bleeding that occurs, and in that situation, the mother is going to start to make anti-Rh antibodies. Now that she has anti-D or anti-Rh antibodies present, she can no longer become pregnant with Rh positive babies. This is the reason that they think Anne Boleyn had so many miscarriages following the birth of Elizabeth I. They think that she was an Rh negative mother and that she became pregnant with Rh positive babies. And this was prior to the time that we were able to treat with what's known as RhoGAM. RhoGAM is an injection that's given to mothers shortly before they give birth to their Rh positive babies. And this prevents that formation of the anti-D antibodies. If this doesn't occur, then you do have the mother's antibodies attacking the Rh positive baby's blood, and this is referred to as the hemolytic disease of the newborn, also called erythroblastosis fetalis. This can create an anemic and hypoxic scenario for the fetus, and it can also cause clumping in the mother's blood. And so this can be terminal based on the degree and amount of time that this is actually occurring in. So that's the reason RhoGAM is given during pregnancy to mothers with Rh negative blood. So the idea is that you'd want to be able to take this information and do a short worksheet in class asking you to identify antigens on a particular individual's cells, identify the antibodies that they make, and identify what blood types they can receive in a transfusion.